So first, I'd like to thank Dr. Medhat and his team for organizing this meeting and for the opportunity to speak to you. My topic is related to RNA, a much more famous topic than it was a year or two ago, of course, because of the vaccines. And that is just the first of many applications we're going to see of RNA in the future. But in this talk, I'm going to be more focused on the biological and the scientific significance of RNA and the unique properties and the unique roles that it plays. I'll exemplify it with a class of RNA called Riboswitches. And one of the distinctive things about Riboswitches, and one of the reasons why it's such a good model system for RNA, is because of the role played by conformational changes that are induced when they bind to other molecules, when they bind to ligands. And in order to be able to make use of and eventually sometime in the future fully exploit these properties, we need to be able to understand the function and therefore to be able to predict the conformational changes before the ligand binds. And that is in some ways the final goal of this work. So I start with RNA, and we need to put it in the context of the central dogma of molecular biology. Historically, this was stated in the following way, that DNA has all the instructions for everything that takes place in the cell, and protein instructions. And in biology, we say function is very closely related to structure. So the implication, if DNA has one function, which is to carry the instructions, that is, to hold and transmit information, then it should have basically one structure. Whereas since each protein has a different task, each of them should carry out a particular function with a very specific structure. And maybe this is one of the reasons why structural biology has mostly focused on the structure of proteins. And nucleic acids have been considered less interesting. RNA initially was thought to be important only because the DNA had to be preserved going from one generation to another. And so RNA, it was thought, was needed as a kind of disposable version of the DNA. So the RNA would be made from the instructions in the DNA, and then those instructions would be read by the protein, and then it would be thrown out. So it was kind of a passive role being played by the RNA. And and in terms of structure, it was thought that the function of the RNA was not very diverse, and so the structures wouldn't be very diverse. And this relationship between structure and function in nucleic acid was exemplified by the base pairing complementarity that we find in nucleic acids. So nucleic acids have 4 bases, G, C, A, and T, or the last one is replaced by U in RNA. And every time we have G, it pairs with C. Every time we have A, it pairs with T or with U. And as a result, if we have a complementary strand of a nucleic acid that has to base pair with the first strand, then the sequence of bases in the second strand is determined by the sequence in the first strand. So this is the way that nucleic acids have been designed structurally for their function of conveying information. But there's a problem with taking that logic too far with the RNA, because the RNA, unlike the DNA, inside the cell almost always is in a single strand. So there's not a single complementary strand. Every G in the RNA has a choice of all the C's to pair with. Every A has a choice of all the U's to pair with. So if we have an RNA that, say, has 100 or 200 or 1,000 or a few thousand nucleotides, which is not unusual for an RNA, then the number of possible base pairing combinations becomes astronomical. And so if the RNA has the capacity to form so many structures, maybe some of those structures could be quite complex in 3 dimensions. So here, for example, we have a representation in 2 dimensions of the small subunit of the ribosome from bacteria. We have, in this representation, the base paired regions represented by these ladder steps. And where we have these base pairs, we can imagine that the structure is going to be something like the double helix. But look, we have a lot of other spots where we have these bubbles, these unpaired regions, and we have terminal loops, and we have branch points. And these could be forming a lot of different structures in 3 dimensions. And in fact, when the 3-dimensional structure of this ribosomal RNA was solved in 2000, and that led to part of the Nobel Prize in Chemistry in 2009, this is what it looked like. It's a quite compact structure which resembles some of those protein structures. So if RNA can form a variety of complex structures, that implies it could carry out a variety of complex functions. Now, another hint as to this much expanded role for RNA came. So this just shows a very brief summary of a number of the different, just small sampling of the new categories of non-coding RNAs that have been discovered in the last 20 years or so. And perhaps the most famous are the interfering RNAs, the silencing RNAs, and the microRNAs. These are RNAs that, in partnership with proteins and DNAs and other RNAs, selectively switch genes and the expression of genes off and on. And this takes place temporarily. So we have certain times in the cell, you know, all the cells in all our bodies, and also in all of them have the same DNA, the same DNA we got from our parents, same DNA that our children will have. And this is the same for the cells in the eye or in the skin or in the heart. But they are not carrying out the same instructions, even though the DNA is the same. So with these non-coding RNAs, we're starting to understand why. We're starting to understand how it is that RNA plays this role where we have cell-specific expression and we have expression that's temporary. So in other words, it may respond to the immediate needs of the cell, and it's involved with sensing the environment of the cell and the needs of the cell and then making a decision in terms of gene expression. So we're starting to come to this picture of this role for RNA that combines its capacity for information storage with its capacity for diverse functions and carrying out diverse roles. It needs to detect information about the cell state and then use that information to make genetic decisions. So we have chosen the Riboswitches as a model system, and we're going to see that it's an excellent model system for these peculiar biological properties of RNA because it's extraordinarily simple. Riboswitches are found primarily in bacteria. What is a riboswitch? I will illustrate with an example. This is the riboswitch. It's called S-adenosylmethionine riboswitch, or SAM riboswitch. It sits in what's called the 5' untranslated region, so upstream of genes that are involved in biosynthesis. These genes code for proteins that are involved in the synthesis of SAM. When there's lots of SAM in the cell, the SAM binds to the riboswitch. That triggers the formation of a structure on the left here called the aptamer, which brings together 4 helices. It's a 4-helix junction. And this structure then triggers the formation downstream of a hairpin loop here that's called T for terminator, because it terminates transcription. So this is a form of feedback regulation. when the cell sees that it has enough SAM, the riboswitch detects that information and it turns off the production of SAM. If there's no SAM around or low levels of SAM, we get the formation of the structure on the right. This is called the transcription on structure because this red part of the terminator is sequestered by a different hairpin called the anti-terminator, AT here, because it prevents formation of the terminator and it prevents termination of transcription. Riboswitches have drawn a lot of interest as antibiotic targets because, as I mentioned, they occur primarily in bacteria. And they are targeted by small molecules. These are examples of some other small molecules that are related to riboswitches. They have kind of drug-like properties. But riboswitches also present a lot of potential for nanoengineering and synthetic biology. From the biological point of view, what's interesting about them is that they are combining these quintessential RNA roles as biosensors, as detecting information, and as gene regulators. So structural biologists have been interested in the riboswitches, but there's a big problem because we can see that these are very dynamic structures, and if you want to define a structure to high resolution, it's hard to do that with a structure that's moving around. So crystallographers cut the riboswitch. They cut off this part that forms the terminator and force the riboswitch into one conformation, forming the aptamer. And this shows a picture of the X-ray structure of the aptamer, the first X-ray structure of the aptamer. There were several that came after that that are similar. And we see that we have the 4 helices. 2 of them are color-coded here in blue, and they stack collinear. And the other 2 are in green, and they also stack collinear, but one of them has a big kink in. And the residues, most of them near the junction that contact SAM, are highlighted in red. So they're highlighted in red here, and the SAM is highlighted in yellow. So it's a compact protein-like structure, and it's performing this function of ligand recognition, of biosensing, which is addressed by the X-ray structure. However, riboswitch function is not just about ligand recognition. If it was, then we would be able to predict exactly when the change in gene expression would be triggered from the KD, from the binding affinity, because that would tell us the threshold concentration of the ligand at which we would have mostly bound RNA. But in fact, we find that with the SAM riboswitch and with other riboswitches, there is not a correlation between the binding affinity and the threshold levels of regulation. So there's something else happening here. And there's also something very interesting happening physically because you have a very small molecule, 400 molecular weight in the case of SAM, rearranging a molecule, conformational molecule, that's 100 times the molecular weight. So in order to explain these things, we have to take account of the expression domain. So last year, I showed you guys this slide when I made a presentation in this meeting. And I talked about how in structural biology, we have these nice, pretty pictures of the different components of the cell, not just RNA, but proteins, DNA, lipids, and so forth. But we can kind of give people the expression, well, first of all, these molecules, they don't operate on their own. They always operate in interaction with each other. And we would think, from the traditional structural biology point of view, that they could fit together like Legos. It would all be about shape complementarity. But that's not true, because these molecules are very dynamic. And I talked last year about how, fortunately, there is a way that we can understand these changes in conformation when biomolecules interact with each other. And that's through what we call the free energy landscape. So let's talk about the free energy landscape. First, for an individual biomolecule, it could be an RNA, could be a protein. We can imagine that that molecule can form a very wide variety of conformers. And we could map all those conformers in a space. Actually, it would be a space very hard to visualize because it would have many, many dimensions, hundreds or even thousands of dimensions. But in order to visualize it, let's imagine mapping it onto 2 dimensions. Each point in this plane corresponds to one conformation for the RNA. And for each of these conformations, we have a free energy. Here we represent the free energy in the 3rd dimension. This gives us a surface. And if we imagine the protein or the RNA on this surface, they would be like we would be if we were, let's say, a skier on a surface like this. We would always tend to slide down towards the lowest points. But we wouldn't just sit at the lowest point, because if we were moving, if we had some kinetic energy, then that would tend, once we got to the bottom, to have our momentum bring us back up. So we would spend most of our time towards the bottom, but we would also sample the high energy states. And we can calculate, actually, exactly the probability that at any given moment, the RNA will be sampling a particular conformer through the Boltzmann distribution. So the probability of that particular conformer will just be proportional to minus the free energy of that conformer divided by the thermal energy. So now this is when the RNA is on its own. Now when the RNA binds to the ligand, for each of these conformations, we have to change the free energy because we have to add the free energy of binding. And sometimes we tend to think that the ligand just binds to one structure, but actually, realistically, there will be a different amount of binding free energy for the different conformations. So we have a kind of binding energy landscape, and that corresponds to the free energy landscape. When we have the ligand bound, the total free energy is the sum of the 2. The free energy, the original conformation free energy, and the binding free energy. And so when we sum those 2, the regions of lowest free energy may differ from what they were before we added the ligand, and therefore we expect a different distribution of conformations. So in order to be able to predict then the conformation when ligand binds, we need to have the binding energies for the different conformations. We need to have the binding energy landscape. But this becomes a problem, because when we measure the binding energies in the lab, then our RNA or our protein is actually in a Boltzmann equilibrium. And our measured binding affinities are an average of binding affinities for the different conformers. So in order to measure the binding energy for individual conformers, we would have to isolate those individual conformers and fix the protein in the individual conformer, which is almost impossible for protein in general. However, for an RNA, if we just talk about the secondary structure, that is the base pairing pattern, then we can actually define a finite number of those, the base pairing patterns, and there are base pairing rules based on empirical measurements that we can use to predict, to estimate the free energy for those different conformations. And of course, in doing so, we're just exploiting these base pairing rules, which we have at our hands for RNA, but not for proteins. So Wei Wang, who was my graduate student at LSU, teamed up with a colleague there, Juyeon Kim, to do those calculations. And it just starts with the Boltzmann factor and then statistical mechanical methods in order to derive the free energy landscape. And we get an idea of the most probable conformers in the absence of SAM. So these were predicted, and then Wei did a number of experiments using susceptibility chemical cleavage, fluorescence, and NMR to validate the presence of these lowest energy conformers. And So then we have the question of the binding energy landscape. We have the free energy landscape of the RNA acetone. We need the binding energy landscape. And this is not simple because in principle there are a tremendous number of conformers that we need to isolate and fix and measure. So what we need to do, we can make a guess that most of the conformers won't bind to the ligand at all. And so then what we need to do is we need to guess which conformers do have some binding and then isolate and fix those and measure their binding affinity. Well, in the mechanism, conformational change mechanism that we saw before, there's a key role played by this red strand. So we see that it can either pair with one partner and give us the P1 helix or pair with an alternative partner and give us the anti-terminator helix. But if we have the possibility of these two different helices, we also have the possibility of what we call these hybrid forms, where we have, let's say, some base pairs forming in the P1 and other base pairs forming in the anti-terminator. And we hypothesize that these hybrid forms that have just at least a few base pairs in the P1 helix can bind to SAM, because the X-ray structure showed that these structures would have all of the elements that are needed to actually contact SAM. So Vamsi Boyapati, another one of my LSU students, used equilibrium dialysis to test this hypothesis. So he truncated the riboswitches in order to fix it into different conformers, either conformers that didn't include the P1 helix, or conformers like the aptamer, which don't include any AT helix, or conformers which contain some of both, the P1 helix and the AT helix, but with different amounts of each. And then he measured the binding affinity. Now, here we have the KD. So remember, with the KD, the lower the KD, the stronger the binding affinity. And that means we have, when we look at this chart, the best binding affinity for the aptamer structures which contain no AT helix. And we have virtually no affinity for those structures that don't contain any P1 helix. But for the hybrid structures, we have, as we hypothesized, an intermediate binding affinity. And from these measurements now, we have the binding energy landscape. So at Zewail City, Osama El-Aidi combined these two, the predicted free energy landscape and the measured binding energy landscape. And he did calculations of the perturbed free energy landscape in the presence of SAM binding. So here we have a representation of the free energy landscape. You know, as we said, we can't plot all the conformations, so we reduce the conformational landscape to a few key conformational coordinates, or reaction coordinates as they're called. So in this representation, we have on the vertical axis the number of AT base pairs, and on the horizontal axis the number of P1 helix base pairs. And the lowest free energy is shown in blue, and we see that that's at the upper left. That means that we have little or no P1 helix base pair formation, and we have long AT helix formation. So in other words, structures like this. And then when we add the ligand, we see a shift towards these intermediate structures here. And the bluest point now is at the lower right, which corresponds to these structures that have almost a full P1 helix, but very little or no AT helix. So these results now show us that, as we hypothesized, the binding free energy is intermediate for the hybrid conformers, and we can calculate the perturbed free energy landscape, and it's shifted towards the intermediate conformers. And there's also an increased minority of what we call the aptamer transition state. This is the one that contains no AT helix at all. And that's important because if there's no AT helix, that facilitates, as the transcription of the RNA is completed, the formation of the terminator hairpin, which terminates transcription. So now we need to look for experimental evidence where we can directly see these different RNA conformations and see the distributions change as we add ligand. In order to do this, we've again teamed up with my former student Wei, who's now research assistant professor at Case Western Reserve University in Cleveland in the United States. And he's now doing cryo-electron microscopy. So we sent him some of our Riboswitch RNA samples, and he has immobilized those RNAs as particles on a cryo-EM grid. And some representative image is shown here. And we see these individual particles. And when we blow up those individual particles, we observe that there are a number of them that are forming a compact shape. It's tempting to speculate that these compact particles are forming something like this aptamer structure. And at the same time, we also have other RNAs here that are forming more extended structures. So Wei has clustered these together, taken the compact structures and taken the extended structures, and using the sophisticated algorithms that cryo-EM people use in order to analyze these different individual particles and combine the data from different individual particles that are oriented in random directions. He's reconstructed the shape of the compact form and of the extended form shown at the top. The resolution right now of these maps is a little over 4 angstroms. That's not enough to see individual atoms, but at least we can see the crude shapes. And it's tempting to speculate that this extended form contains the anti-terminator and the compact form contains the aptamer structure along with the remaining part of the riboswitch. Consistent with this speculation, Wei has also looked at this construct where we've truncated a good part of the P1 helix. So we force it to form this long AT helix. And then what we see is a lot of extended structures. There's still a few of the compact structures, and this is what we would expect. We always expect an equilibrium. But having perturbed the system towards the AT helix, as we expected, is perturbing us towards the extended structures. So this is very early days, and we'll see. We're still working on trying to get higher resolution data and getting data now for cryo-EM on some of our other constructs. So finally, I'd like to thank people involved in this work. First of all, Wei. Of course, he did much of this early work with the calculations and the experiments on the RNA on its own, and now more recently with cryo-EM in his current incarnation at Case Western Reserve, Vamsi Boyapati for the measurements of the equilibrium dialysis binding affinities, and also Juyeon Kim and Shantanu Jha, our collaborators there at LSU, and also Osama for his work doing the calculations of the perturbed free energy landscape, and then our team at Zewail City, Yosef Mansour, Abdullah Abdel Sattar, and Fayrouz Mohammed in particular, who've been doing the cloning and the production of the different RNA constructs, including those that have been sent for cryo-EM. And finally, I want to thank all of you for listening.